Hello, welcome to today's lecture. Today we are going to talk about the complement system. In last section, we have introduced the antibody. It is an important molecule in the serum. But in fact, there was another important molecule in the serum, which is called complement. Let's start with such an experiment. This is a serum. It is a fresh serum containing the antibody against the cholera. When the serum was incubated with the cholera at 37 degrees, and then the bacterial lysis can be observed. But if the fresh serum was heated to 56 degrees for 30 minutes, and then it was incubated with the cholera again, then the bacterial agglutination will be observed instead of the bacterial lysis. So what can we conclude from this experiment? The first one, antibodies are relatively heat stable. As we know, the bacterial agglutination, it is a complex of the antigen and the antibody. Those antibody and antigen complex can be visualized in vitro as the agglutinate. When the serum was heated to 56 degrees, that means the antibody combined with the antigen. The antibody still combined with the antigen. That means the high temperature will not destroy the structure of the antibody. So the antibody combined with the antigen to form the complex. But the antibody itself cannot lead into the lysis of the bacteria, because when it was heated to 56 degrees, we cannot observe the bacterial lysis. That means a second component in the serum will be responsible for the bacteriolytic activity. And that component is heat sensitive. It was called complement. A scientist, Bourdet, was awarded the Nobel Prize for such a discovery. In fact, the complement is not a single molecule. It is a complement system. It contains a collection of soluble proteins present in the blood and other body fluids, such as the C1, C2, C3, and C9, and some soluble factors, factor B and factor D in the serum, as well as those cell-bonded proteins, for example, complement receptor. So in the normal state, the complement exists in an inactive state. But once activated, they interact in a highly cascade to carry out a number of basic functions. There are totally 3 pathways to activate complement system. There are classical pathway, alternative pathway, and lectin pathway. Today we will focus on the classical pathway. The classical pathway is initiated by the binding of antigen-antibody complex to the first component of the complement, C1 molecule. And C1 molecule is composed of 1 C1q molecule and 2 C1r and 2 C1s molecules, as shown in this figure. This is the C1 molecule. And C1q is look like a flower of tulip with a stalk and 6 heads. When the C1q bind to the antigen antibody, it can activate another 2 proteases, C1r and C1s. The head of the C1q combined with CH2 domain of the antibody. It is the Fc region of the antibody. So after binding, another 2 proteases another two proteases, C1R and C1S, will be activated. After their activation, C1R and C1S will cleave their substrate, C4 and C2. The antibody involved in the activation of the classical pathway are IgM and IgG. But soluble antibody cannot activate complement. Only antigen-binded antibody can activate C1q. Let's look at this picture. So this is soluble IgG. At least two Fc portions of the antibody bind to the C1q, then C1q can be activated. As we know that soluble IgG is in form of the monomer. So that means IgG only have one Fc portion. So soluble IgG cannot activate C1q. But once the IgG bind with the antigen and two adjacent IgG combined with C1q, so C1q can be activated. And another soluble IgM, as we see here, IgM is in a pentamer. It's made of 5 monomers. So C1q combined IgM. But unfortunately, soluble IgM exists in the planar configuration. That means the Fc portion was hidden and is not available to binding for C1q. And after binding with the antigen, IgM exposed to Fc portion, allowing the binding of C1q. Then C1q can be activated. So soluble antibody cannot activate complement, and antigen-binded antibody can activate complement. In addition, at least 2 molecules of IgG combined and activates complement, and 1 molecule of the IgM can activate complement. Based on this point, IgM is a more efficient complement binding antibody than is IgG. Let's look at how the classical pathway is activated. This is a pathogen expressed multiple antigens. Their antibodies bind the antigen to form antigen-antibody complex. Then the first component, C1q, bind Fc portions of the antibody to activate C1R and C1S proteases sequentially. The substrate C4 and C2 will be cleaved into pieces respectively. The small fragments of C4a and C2b are released. The big fragments of C4b and C2a attached on the surface of the pathogen are called C3 convertase. C3 convertase then cleaves C3 molecule into fragments. The small fragment of C3a is released, and the large fragment C3b will be attached on the surface of the C4b and C2a to form C5 convertase. Finally, C5 convertase cleave the C5 molecule into C5b and C5a. C5a is released again, and C5b will bind C6, C7 to form C5b6-7 complex. C7 inserted into the surface of the pathogen, but it is not stable. C8 also binds C5b6-7 complex and insert into the cell membrane. Multiple C9 molecules were recruited to make a pore on the surface of the pathogen, finally leading to the death of the pathogen. Therefore, the terminal sequence of complement activation involves C5b, C6, C7, C8, and poly C9, which is inserted sequentially to form a macromolecular structure, and then inserted into the membrane of target cells, leading to the cell lysis. This macromolecule is called membrane attack complex, MAC. This is the activation of classical pathway, and the terminal sequence of the classical pathway is also a common step for all three pathways. That's all for today. Thank you for your attention.